Good morning, everyone. I'm honored to open up the Linné lectures today after the 3 Celsius sessions yesterday. And today we'll switch in the first session to chemistry. And for the audience on Zoom, or online, I want to mention that in case you identify a delay between picture and sound, please restart your browser and then it should be synchronized. Okay. So I'm really honored to have Jeremy Nicholson here on Zoom with us, who will give the Linné lecture. And just a few words about him. He actually had a PhD in biochemistry, and he was a full professor of biological chemistry in 1992, which was, I assume, one of the first. He then had several chairs at the Imperial College of London, and he founded and became director of the world's first National Phenome Center at Imperial College. Afterwards, he, in late 2018, he became a pro-vice chancellor for health sciences at Murdoch University in Western Australia, where he's now directing the new Australian National Phenome Center in Perth. And so just about his accomplishments, he has over 850 publications in different areas among analytical chemistry, metabolomics, microbiome, and systems medicine. And he holds 12 honorary professorships at different universities. I think that's also really an accomplishment. Has over 76,000 citations, and also with these accomplishments, of course, several awards come along, and I just want to mention one of them, which is like the Lifetime Honorary Fellow of the Metabolomic Society, which was the first handed out. And now we're looking forward to your lecture. Please take over.
Thank you. Well, good morning, ladies and gentlemen. I'm broadcasting to you from a very sunny and hot Perth in Western Australia. I think it's 39 Celsius outside. Have you got my screen up there? Okay, you should have the whole lot now. Okay, well, firstly, thank you very much for inviting me to give this lecture. It's a great honor to do it. Of course, as part of it, I've done a little bit of boning up on Linnaeus and Celsius, learned a few things that I didn't know before. But of course, Linnaeus, very famous for all his organizational papers about bringing order into biology. And of course, the binomial system of classification of organisms is still used, and you could argue that Darwin needed that sort of background information to be able to move on with his own work. And in many ways, Linnaeus is similar to later workers who, like Mendeleev, who classify the periodic table. So bringing order to science is necessary for progress. But I think that Linnaeus now, if he were to see us today and look at all what modern genomics can do, I think he'd be fairly surprised. And of course, the world isn't really Linnaean in the way that he would like to classify it. Things are much more of a continuum, and there is actually a lot more flexibility in nature than probably Linnaeus or indeed Darwin thought. What am I talking about? Well, I'm talking about the sort of the continuum of everything from hosts and microbes to metabolism and immune systems, and how that all works together to create who we are as individuals and what our likely diseases are going to be in the future. And potentially through understanding those interactions, we create new knowledge that will allow us to better handle those diseases and stratify patients and give the right treatment for the right patient. So we set up a laboratory here in Western Australia. You should be able to see part of it. It's right next to one of the main hospital complexes, one of the most advanced hospitals in Australia. And it's a suite of advanced spectroscopic laboratories with NMR and mass spectrometry. There's about 12 NMR spectrometers and 30-something mass spectrometers, all focused on understanding complex metabolic processes. And I'm going to explain now how we apply that sort of technology and approach to understand the basis of disease and what we're going to try and do about it in the future. So firstly, remember everything comes from genes and environment interacting in complex ways, continuous stream of interactions throughout life. And genes and environment determine who you are and who you are going to be. And at the same time, they also determine your metabolic composition and your metabolic profile. So if you want to understand the metabolic profile, it's telling you something about the gene environment interactions. And the other thing to remember is that everything, every part of your body, the surfaces, the inside of your body, your guts, are full of bugs. And the interaction between genes and environment occurs through those extra genomes. And out of all of this complexity emerges the phenotype, the phenome. And if you think about it, and you go back to Darwin and you go back to Linnaeus, I mean, everything they were doing was actually phenomic properties that they were working on. So microbes are also associated with lots of different non-infectious diseases, as well as the infectious ones. And the microbiome itself strongly modulates the immunometabolic interactions in the body. Quite a lot of what you are metabolically is determined by your microbes and what they're doing. And disorders of the microbiome, and that means abnormalities, whatever that actually means, and that's quite a complex definition, but they're associated with a huge number of different types of disease. And you see some of them listed here. There's inflammatory diseases, there's cancers, there's gut diseases, of course, diabetes, etc., etc. So, in addition to our own genome, the variations in our own genome influencing our probabilities of getting disease, there is interacting with that in real time another set of genes which are inside us. So whenever you're thinking about personalized medicine, you have to take into account the fact that we are supraorganisms. We are bigger than one organism, and a lot of the metabolic control is actually devolved back to bugs, not just the human genome. So we think of what we call metabolome, co-metabolome interactions. So here, I'm trying to represent the fact there's a whole series of cell types in the human body. There's actually about 500 different cell types in the human body, and they've all got their own different transcripts and proteins and metabolites, and they interact in real time with the gut microbes. And the microbes modify foods and catabolize them. They make molecules of their own, which is to do with signaling and controlling their own biology, quorum sensing as it's known. And those molecules also cross the gut barrier, enter the body, and are further metabolized, and they also hit a whole series of receptors. So those metabolites that are made by the bugs aren't just passive, they're actually active, involved in the symbiotic control of metabolism. And then things that are made in the human body are also secreted back into the gut, and we end up with a sort of enterohepatic complex circulation of biochemistry, which involves creates combinatorial chemistry. A good example of that is from bile acids. So if you've got something like cholic acid, which is made in the liver, it's glycine and taurine conjugated to create the bile acids are involved in digestion of fats, emulsification of fats. They go into the gut and they're partly deconjugated by the gut microflora, particularly lactobacilli, to regenerate cholic acid, which can then undergo 7-alpha dehydroxylation to create deoxycholic acid. Deoxycholic acid is a combinatorial metabolite. It takes more than one genome to actually create it. And that can then be reabsorbed and has loads of other metabolic reactions as well. But importantly, deoxycholic acid, like many other bile acids, are biologically active. And it was shown about 10 years or so ago that in obese people, then high levels of deoxycholic acid that are produced by the abnormal microflora is related to the generation of cancer, and particularly liver cancer, but maybe also colorectal. So here is the interesting thing, is that things that are made in your body can be modified by bugs to change your risk of getting disease. And I think that's a pretty new concept. We've known about the gut microbes for a long time, but we've only realized, or beginning to realize, about their importance in our biology and our diseases in maybe the last 15 or 20 years. If you go into a little bit more detail and look at germ-free animals, for instance, versus normal animals, so germ-free, these are rats that were born by cesarean section, and you do a bit of liver transcriptomics, you find that a lot of the enzymes involved in bile acid metabolism, cholesterol synthesis, and steroid biosynthesis are all modulated by the microbiome. And everything in red are enzymes that are in the human body that are modulated by microbial signaling. So again, the depth of which this operates has not been fully appreciated until relatively recently. So anything that goes into the body that upsets the microbiome is likely to have some short-term consequences, but potentially also long-term consequences relating to disease risks. And here are some of those processes or pathways which actually have drug interactions. So statins act around here, and then there are cytochrome P450s, some of which are druggable. So there is a drug-bug interaction that's intrinsic to us. If you think about this, about the whole of the gut, there's lots of different bugs making different metabolites at different times, hitting different systems. So there are metabolites that hit bile acid nuclear receptors. There are endocannabinoids, and they hit the endocannabinoid system tone axis. There are aromatics of various sorts, the short-chain fatty acids, and all of these are being produced at the same time, together by different organisms. So we call this a microbiome orchestra. It's playing a complex tune of metabolites which are being listened to by the various receptors in your body which are responding to those metabolic signals. And as you can see, this goes all the way up to leptin and satiety detection, and the brain as well. So it actually works all over the body. So that's complex, but of course it does mean that there are lots of new druggable targets. So I think Pfizer calculated about 15 years ago, there are about 3,000 druggable targets in the human genome. Well, if you look at the microbiome, there are a vastly greater number of druggable targets. So there's a potential for a whole new industry for the future if we really understand this. So all of this creates complexity, and that means complexity of metabolism, which we really have to start trying to understand. So let's talk about some of these things. So firstly, the idea of using metabolism and metabolic processes to understand disease is not exactly new. And this is representative of some 16th century work. And Ulrich Pinder published this book, The Inevitable Fate, back in 1507, 200 years before Linnaeus was born. And on the right here is a rather neat little chart, which has got pots of urine and their smells and tastes and etc. listed them in relation to potential diseases and also therapies. So this is actually conceptually is a sort of chemical pattern recognition diagram where all of those features are related to some actionable path that a clinician would take. Now, fortunately, these days we don't have to taste urine anymore because we have spectroscopic tools to do that for us. So the most important ones for metabolic phenotyping are NMR and mass spec. And that's because they give information on hundreds or thousands of metabolites in a single analytical run. And in NMR, there's loads of different experiments you can do to pick up different information from the same sample using a whole range of different radio pulse sequences. And in mass spec, there are different chromatographic tools you can use on the front end and lots of different sorts of mass spectrometers with different sorts of ion optics designed for separating certain sorts of molecules. And the important thing about this is that every single technique you use gives a slightly different view. It has a different window on the metabolism that you're looking at because it picks up different things. So when you're trying to do comprehensive metabolic phenotyping, you have to use combinations of technology in order to get the answers you want. And on top of that, there are literally thousands of statistical tools and multivariate statistical tools for helping you interpret the data. There's chemometric modeling, which includes pattern recognition, classification. In fact, Sweden is one of the world centers for that. Umeå University is very famous for that. Svante Vold was there. And statistical spectroscopy, where we're using a range of different statistical techniques for mixing together different spectroscopic data sets to be able to co-analyze what different techniques are producing from the same samples. I'll show you that a bit later. It's very powerful to do that. And there's also the use of statistical thermodynamics as well, which basically to look at entropy and disorder of metabolic systems based on understanding of the various concentrations there. So this is almost the opposite end to pathway analysis. This is looking at the total disorder or specific pathway disorder using thermodynamic principles. I'm not going to go show you all the toys we've got in our lab, but basically the journey in our lab starts in the NMR facility. We have 8 600 megahertz NMR spectrometers. 6 of them are set up for in vitro diagnostics. So each of these machines can run 30,000 samples in full automation a year. So instantaneously, you have high throughput technology. Capable of dealing with very large numbers of samples. We also can do a whole range of different experiments, and we typically do about 6 different NMR experiments on every sample. So you're already doing with big data, even just on the NMR side. And in mass spec labs, we've got multiple machines of various configurations for running different types of analytical procedure. And if you put it all together, this is a simplification, but we measure typically lipoproteins and a range of low molecular metabolites by NMR. We do quantitative mass spec lipid screening. We measure a whole range of inflammatory markers, chemical markers via mass spectrometry. We do amino acids and tryptophan pathway analysis. We do bile assays, and there's multiple targeted assays. And we also do the immunology. We do cytokines. We do exploratory techniques like 4D lipidomics. We do untargeted Hillick, UPLC, QTOF measurements for polar molecules, and we have the equivalent for non-polar molecules. And we also have things like direct injection Fourier transform mass spectrometry. And that is what we apply to every single sample in our lab. So we end up with a huge amount of data per sample. And the way we think about deploying these technologies, there's 2 different sorts. One is from the point of view of population analysis. So here we're interested in what epidemiologists are interested in. We're interested in finding signatures that detect at-risk groups. So can we do a blood profile of somebody and know what the likelihood of them getting a stroke is in 5 years or getting cancer or something like that? So that's a prospective type of analysis that we try to do for large-scale phenotyping studies. And it's to inform future healthcare policy. In the clinic, we can use exactly the same technologies, but we're on a different timescale. We're on the clinical decision-making timescale, and that can be very, very quick. If it's a surgeon, it requires second decision-making. But if you're in intensive care, that might be a few hours to make a decision from the sample going in to being analyzed and you doing something about it. So, and of course, the outcomes of the experiments there are very determine what the doctor does next. So it's very important to align the analytical stuff with the clinical decision-making timescale. We created a Phenome Centre network, and this is part of the reason I'm now in Australia, because I ended up in one of the labs that we started building. The climate's just nicer here. There's other reasons as well. But the Phenome Centre network has harmonized technology, and there's a whole series of labs across the world. So if we do something in our lab, we can immediately compare it with something that's done in the UK because we've done the same thing. So harmonization and standardization is an important part of what we do. And together we're working on all the major unmet medical needs from around the world. And here are some of the ones that are well known: diabetes and aging and dementias and neurological disease. This affects everybody. We all have a common interest in doing that. But of course, different countries have got different gene environment interactions. So the expression of a particular disease varies a little as to where you are around the world. And of course, since 2020, COVID-19 has become a really important subject for us all to study. If I just take you back to from the epidemiology point of view, we actually been working on epidemiological samples for a number of years, but the most important paper that we produced was basically the construction of the metabolome-wide association study, where we're looking at metabolic profiles and then trying to relate that to population variation in metabolism and or disease outcomes. And if you to look at this as sort of one of our first studies with 4,600 and something people sampled twice, looking at the urinary metabolome, it's quite interesting. All these different people in different parts of the world, you can stratify them and classify them as to where they came from based on their urinary metabolic profiles. And if you were to look at that in relation to just sort of generalized mortality statistics and BMI, cardiovascular disease, etc., it pretty much goes in that direction. And people live in southern China live relatively long, healthy lives. That's because historically they've had healthier diets more than anything else. And people that live in the sort of the southern states of America tend to live less long and have less healthy lives. And so who you are and where you are in the world can be related to your metabolic phenotype. And it's more than that, because where you live also determines what you're going to die of. So if you were looking at the Japanese Americans, and we had a few hundred Japanese Americans in this study, they don't map with Japan, they map somewhere near Baltimore or Minneapolis or something. So once you live in a country, you tend to have the same diet and lifestyle, and you get the same diseases as them as well. We take that on a little bit further, and you can look at, for instance, human obesity, although it's fashionably called adiposity now. And we were looking at various biomarkers that related to BMI. And then, again, these are urinary biomarkers, but we can look at plasma as well. And this is just the top 20 most discriminating biomarkers that relate to obesity profiles. And the ones in red are actually ones that have substantial gut microbial involvement. And what's interesting is if you look at the urinary microbial metabolites and their variance in relation to population BMI, it explains about 8% of human BMI population. And that doesn't sound like very much, but it's a lot better than the genomics, which in the biggest ever GWAS study on BMI, published in Nature Genetics, about 250,000 people, the total BMI was only explained to the variance, only explained to level 1.45%. So the microbes are actually much more important at the population level to do with the development of obesity in the general population. And if you look at some of the pathways involved, this is now taking those data and re-expressing it in a what we might call a network format using something we constructed. It was actually Dr. Yoram Hosmer, who is a lecturer at Imperial. He built this network, and it uses the KEGG network as the basis of it. And every one of these lines represents a single enzyme conversion. And the software is arranged such that when you feed in the biomarkers, positive or negative, associated with a disease process, or in this case, a pathophysiological process, it reconstructs the virtual KEGG network that connects them all together. But it does it in a way that minimizes the number of enzymatic conversions that connect the biomarkers together. So everything in red is positively associated with BMI. Everything in blue is negatively associated with BMI. And all of the coloration that's in here is basically to color coding the different parts of metabolism according to the major sites in which those metabolic transformations are going on. And you can get a little bit cleverer than that. You can actually add in some microbial genomics, so you can look at microbial islands within human metabolic space. So this is a symbiotic metabolism diagram. And this is the sort of thing that would have probably confused Darwin and even Linnaeus, because it's actually looking at the metabolism and the biology of two or more than one organism together at the same time in an integrated format. We can also look at interventions to see how diet changes, for instance, your microbiome activity and potentially disease risk. So this is a study we did a few years ago, and we had noticed that African Americans have got something like 30 or 40 times the colorectal cancer risk as their African cousins. And of course, there are strong genomic similarities between African Americans and Africans. And therefore, it is more likely that it is diet or environment that is causing these differences in colorectal cancer risk. So we did a fancy study where we took a lot of Africans and we gave them American diet. So we had burgers and chips for a couple of weeks. And we took a load of Americans and we gave them porridge and cereals and stuff like that, healthy stuff, which they really hated, by the way. And then we looked at their microbiomes before and after diet. And cutting a very long story short, they did a crossover. So within 2 weeks, African Americans became Africans and Africans became Americans in terms of their microbiome structures. And also the metabolism shifted in dramatic ways as well. And if you then also, these lucky people, we did colonic biopsies on them as well. And we were able to look at some of the cancer risk cells, so things like CD8 markers as well. And they also inverted. So you can show that microbiomes are responsive to diet, they're responsive to healthy diet, and your potential risk factors can be changed in relatively short periods of time. And I think that this is a message that everybody really needs to be able to think about carefully, because you can really change your long-term disease risks by some sensible dietary modifications. Oh, that's the Metabo networks diagram that goes all the biochemistry to do with that. I'll tell you what, I'm not going to go through that at the moment. It's a bit complex. So moving now from populations into acute medicine and patient journeys, one of the things we need to do is apply our technologies and approaches to try and understand patient biology so that we can optimally intervene therapeutically. And that's what a patient journey is all about. So a patient comes into the hospital, there's a series of diagnostic procedures, it could be ultrasound, they could be blood tests, whatever it is. There's an exact diagnosis is made, and based on that, an intervention occurs. This could be chemotherapy or something like that in the case of cancer. And then in the journey, there is some outcome of the intervention. And so what we started to do at Imperial, and we've continued to do here, is we've got this model of the patient journey, and we take various sorts of samples and we perform all the phenotypic analysis on it in order to get this much deeper picture of the dynamic journey of the patient. And this also lends itself to what we call to phenotypically augmented clinical trials, then where we're looking at this phenotypic evolution through time in the patient journey as information that will help us understand good or bad outcomes for therapy, either on experimental drugs or in the general clinical environment. And this approach is scalable and translatable to almost any type of disease. So at Imperial, we did all of these different things. We're doing a few of these things now, but we've only been here a few years, so we haven't had really time. And by the way, COVID-19 has been in the way. So for the last 2 years, pretty much we have been working on COVID-19 related research, and it becomes an exemplar which we can now use to show how we can apply phenomics in the clinical sense. So phenomic discovery and translation, so using COVID-19 as an exemplar for trying to talk about all the things that we've discussed today already. Back in the 1960s, there were a load of famous films, science fiction films about bug epidemics and pandemics. There was the Andromeda strain, which is pretty famous. This one is particularly good. I like this one because the poster is so great. The Satan bug. Since time began, man has hunted the ultimate evil. Now the search is over. So as far as I'm concerned, SARS-CoV-2 is the Satan bug. It has as many nasty things as I've ever come across in any infectious disease in my life. But it's interesting, the timing of all this stuff, because only today, your Minister of Health said it's all over and COVID-19 is no longer classified as a danger to society. That's an interesting view. I'm glad she's so well scientifically informed. I actually beg to differ on this, and I'm going to explain why now. So today's statistics is 403 million people had it, nearly 6 million people dead. We're in the sixth wave. The number of deaths is climbing again. But there's also a hidden cost to all of this because hospitals are shut down and patient wards are full. There's actually a lot of excess mortality going on, which is estimated as 3.4 times the number of deaths. So if you look at today's statistics, around 20 million people have died already because of COVID. And if you look at that in relation to the Second World War, 25 million military casualties over 5 years. This is 20 million dead over 2 years with COVID, and maybe 70 million dead in total, and including civilians. So the COVID statistics are right up there with the Second World War, and I'm telling you that it is not over yet. From a point of view of the clinical presentation, most people are looking at it from a respiratory point of view, which is not unsurprising. Coronaviruses cause respiratory disease. But this also gives rise to misplaced views and opinions about severity. So Omicron regarded as mild because it produces different respiratory effects. There is no evidence that it actually is mild long-term, nor is there any evidence that is mild physiologically. And if you look at the right-hand side of the diagram, in addition to all the respiratory effects, there's all these multi-organ effects that COVID-19 has, which are actually far, far more severe than you would get with influenza. And so when you are recovering from a disease, are you recovering from the cough, or are you recovering from the systemic changes caused by the virus infection? There's also an evolving legacy of this, the post-acute COVID-19 syndrome, also known as long COVID, and that too affects almost every organ in the body. So if you get COVID, you have a significant chance, and the statistics vary, I'll come back to that in a minute, significant chance of having some long-term effects. It also changes the dynamics of the microbiome, and we've already been discussing the fact that changes the microbiome change your long-term risk effects, and that's on top of long COVID. This is an extra piece of statistics that we really don't understand yet, but it's been in one of these papers that's published very recently. There's persistent respiratory symptoms have been correlated with opportunistic gut pathogens and also the neuropsychiatric symptoms and fatigue, which are associated with long COVID. So the bugs have been affected, and that intrinsically means there's going to be a long-term effect. The way we're approaching this as a research program, you'll have noticed that there aren't many cases of COVID in Australia until relatively recently. We're now at a couple of million, but only about 7 or 8 months ago, we only had 30,000. So we've got samples coming in from all over the world. We have an import license for COVID samples, and we're working with Cambridge and Harvard, Imperial, and we've got about 10,000 COVID samples from multiple sources. We've also got our own epidemiology studies, and what we're trying to do is map the progression of the disease from the normal population state, which has got subclinical disease in it and multiple inflammatory states and also diabetes, et cetera, into the COVID-19 space, where a whole new series of inflammatory windows open up, and then back through the recovery phase, what we might call post-COVID-19 or long COVID. And in fact, it's complex, because what happens is, depending on where you are in that normal population space, you get different levels of severity, you go to different spaces here. And when you recover, you don't go all through the same trajectory. And when you recover back into the general population, you don't go into the place that you were before. So your long-term risk for a whole series of diseases have probably altered forever. And we're only just beginning to understand that. And these processes we call primary phenoconversion, when you convert from the normal, whatever that is, state into the COVID-19 state, secondary phenoconversion when you convert into long COVID, or let's call it the recovery state, and then a final phenoconversion step where you go back, if you're lucky, into the normal population. We also have multiple studies to do that. So we've got the variety of different epidemiological studies, which we've run through or running through here already as part of what the Phenome Centre does already. And then we're feeding in multiple studies from abroad, so the Cambridge severity study, which has also got up to 1-year follow-ups and things like that. And then we're relating that informatically to all the literature that's been collected from around the world. And the patient journey itself is a model that fits in the middle of that. And we've also got pediatric studies from Harvard that we're working through at the moment. So we can feed these in one after another, cross-validate the data between different sample sets. So find out if Japanese COVID is the same as American COVID. It's not, by the way. And find out the different metabolic diagnostic criteria for each of the different subconditions. We're interested in detection and prediction. We're interested in severity and mechanisms, and we're interested in long COVID. So all the technology is designed for picking out those sorts of different biomarkers and panels of biomarkers that give insight into the different subtypes of the disease. One of our earlier studies published last year, this is joint with Cambridge, I'm in here somewhere in the football team of authors, was actually looking at the immunology side. That's really done by Cambridge. And one of the things they discovered is really important is that people that don't get severe disease get early CD8 cytotoxic T cell activation. So what happens is the cells get infected and they send out cytokines and they signal to the CD8 cells to come in and kill those cells where the viral replication is going on. Where there is a robust response to that, a CD8 response, those people do not go on to get severe respiratory disease. However, the actual cytokine response that they get as part of the CD8 process does trigger a series of other processes which are probably related to long COVID. So thinking about this now from the metabolic point of view, let's open all our windows of metabolism. This is an expanded version of what I showed before, and this is what we're doing for COVID-19. I've got nowhere near enough time to take you through all of these. I'll take you through a few that illustrate what's going on. So one of our first studies was actually put a whole series of techniques together and do mixed modeling of the metabolic phenotypes. And here you can see there's controls and COVID, and here is a test set that's been modeled in there using partial least squares discriminant analysis. There's transitions you can observe from people who are in disease through time into recovery. And we can look at all the different biomarkers associated with the COVID disease process, irrespective of severity at the moment. So one of the things we see, for instance, is drops in HDL4. That's very significant. These are small, dense parts of the HDL lipoprotein series. We get change in Fisher's ratio, which is a amino acid ratio, branched chain amino acid to aromatic amino acids related to liver metabolism. We also get changes in things like amino acids, glutamine to glutamate ratio, and we get changes in things like the kynurenine levels and the tryptophan levels. So the whole range of different stuff that we can see emerging from that. If you just take one of those, the changes in amino acid metabolism, kynurenine metabolism, are actually very profound, and they can be linked in directly into the inflammatory cytokine responses. So if we were to look at some of these metabolites, which are markers, again, don't worry about severity. Pretty much these go with severity, but this is a general model. We see a load of things are increased, and we can see a load of things are decrease. So quinolinic acid, there's an interesting one. That is actually a potent neurotoxin in its own right, and it's used in an experimental model for generating Parkinson's disease in rats. So having a high level of that in your blood is not a good thing. We also have 3-hydroxykynurenine and quinolinic acid are also known to be related to Huntington's disease and also Parkinson's disease as well. And things like neopterin, which is right up there, that's related to the cytokine interferon gamma response, which occurs when macrophages are activated. So even in the amino acid pathways, we're seeing all sorts of things of real fundamental biochemical significance that relate to the pathology and potentially relate to long COVID. Looking at it in a different way, here is tryptophan. Blue means it goes down, and COVID red means it goes up. Here is the way that some of the cytokines interact with the indole dioxygenase, which converts tryptophan into N-formyl kynurenine. And here's the other pathway, which leads off to serotonin. The red things go up, and disturbances of this pathway, if you do the literature search, you find that all of these things are associated with this pathway, like HIV, dementia, Tourette syndrome, psychiatric, as well as multiple sclerosis. And interestingly, things like systemic lupus erythematosus, which is autoimmune disease, which has very similar multi-organ involvement to COVID-19, especially with relation to the extreme long-term fatigue. And also, this pathway is related to atherosclerosis. And that's a worrying thing as well, because we've got quite enough of that going on in the world as it is. And indeed, new onset atherosclerosis has now been related to COVID-19 as well. So if we were to look at some of the lipoproteins, which are characteristic also abnormalities in cardiovascular disorders, you see there are strong cytokine connectivities between the lipoproteins, which we measure by NMR, and some of them are particularly revealing. So for instance, this is the apolipoprotein B100A1 ratio. That is a classic marker of cardiovascular risk and myocardial infarction risk. That goes up hugely in active COVID-19 and is related to the CCL5 cytokine that is produced. So that drives the production of this, and this is interestingly is related to the CD8 cells, right? So the CD8 cells, which I've just mentioned, are involved in that early response, also create a chain reaction of biochemistry, which leads to change in the lipoproteins, which changes your cardiovascular risk. So, you know, God is generous. He allows us to do one thing, protect ourselves against the severe injury, but that process may actually open us up to longer term systemic injury as well. So going back to this, so apolipoprotein B100 and apolipoprotein A1, well known associated with atherogenicity. And if we were to look at some of these patients, this is a Western Australian patient, normal patients. This is general population, so some people have got high-ish ABA1 ratios anyhow, they have cardiovascular risk. When they're in the acute COVID phase, they move up in risk. In fact, they move up significantly from low risk up into medium risk during the acute phase, which is what's going on here. And this is from some Cambridge controls and also Cambridge patients who had a much greater range of severity than the ones that we studied in Western Australia. And you can see they're moved higher. And that relates from the mean follow-up. This is follow-up at 6 months. The follow-up period, they're still in medium risk. And the patients who are in the earlier, in the high end up here, are right into up to 10 times the myocardial infarction risk, even 6 months after the COVID episode. So this has really significantly affected our long-term health and will probably continue to do so for years. And so, of course, we're looking at different ways of studying the long COVID, and we have this phenoconversion and phenoreversion approach, where we look at the new phenotypes and emerge. And also interesting, and during phenoreversion, there are emergent phenotypes. There are new abnormalities that weren't present in the active phase of the disease. So it's a very dynamic and complex disease and varies enormously from person to person. This is an interesting one. I'm not going to go through the detail, but this is a healthy cohort of people. This is a COVID positive cohort. This is just back to amino acids metabolism again. And this is some follow-ups. And you can see actually there's several changes here, but indole-3 acetic acid is elevated long-term. And that is a microbial host co-metabolite. And it's metabolic evidence that the microbiome is perturbed. We can look at other types of models. This is one-dimensional OPLS. These are controls looking at the amino acid in a range of patients. And these are follow-up patients 3 to 6 months after they've so-called recovered. These are, by the way, non-hospitalized patients, all right? So that makes this even more interesting. This affects, this non-hospitalization is normal. And you can see only a small number of these patients have gone back to metabolic normality. And the same can be applied to the lipoprotein model here. We've got slightly more higher proportion of people going back to normal on the lipoprotein model, but there are still people out there that are sick. And so we can go through metabolite by metabolite, looking for individual groups of patients. And the blue ones are all the recovery patients. Again, these are non-hospitalized WA mildly affected patients that we can look for panels of cellular immune activation. This has gone back to normal, as you'd expect. Neuroinflammatory problems, there's 3 patients out there that are abnormal. Heart dysfunction, there's a number of people here that are not correct. So we can use this as an objective model for functional recovery of long COVID. And this is, I like to be up to date. So this paper is from the latest edition of Nature Medicine. It's this week's edition, and it shows you the different long-term 1-year cardiovascular outcomes. This is based on 157,000 patients compared to 10 million controls. So the statistics are strong. And what you find for ICU patients is their total or any cardiovascular negative outcome at 1 year is 300 in 1,000 patients. So it's a 30+% increase in cardiovascular risk at a year. And the hospitalized patients are 150 plus, and even the patients who are not hospitalized are about 25 per 1,000. And if you scale that across 400 million people, that's an awful lot of people that are increased cardiovascular risk. One final thing I want to say, which is new markers. Can we develop new markers to detect this? Because basically that paper there said, hey guys, we found all these problems related to long-term cardiovascular risk. What we need now is fast and efficient measures for looking at it in the general population. Well, here's one we prepared earlier. And it's again using NMR spectroscopy. And we use some fancy pulse sequencers using diffusion and relaxation editing to go from the incredible complexity of the thousands of molecules in plasma down to much simpler spectral profiles, which carry key biomarker information. And here, just based on this pulse sequence here, the so-called Dyer pulse sequence, it separates off controls. from the COVIDs very nicely. And there are some important peaks in there. There's a alpha-1 acid glycoprotein, acute flavour reactive protein that comes up here. And this is from a SPIC peak. That's a supramolecular phospholipid composite peak. So it's multiple phospholipids that are embedded in lipoproteins. And it turns out that that is diagnostically tremendous. So if you look at the SARS-CoV positives versus SARS-CoV negatives, that's people with other respiratory disease, it's non-significant. The controls versus SARS-CoV negatives. And if you look at the SARS-CoV negatives versus positives, right? So respiratory disease, it's COVID or non-COVID, 10 to the minus 9, and COVID versus control, 10 to the minus 10. And in fact, the statistics are even better for that on bigger sample sets. We are not satisfied with that being a good enough diagnostic. We're trying to improve it. So this is in last week's analytical chemistry, I think. We've actually improved the pulse sequence. I'm not going to go through the detail, but this is what the one-dimensional experiment looks like for those biomarkers. And this SPIC composite here is obstructed partly by a glucose peak and some other peaks from amino acids. And this GLIC peak here is obstructed partly by CH2 signals that are in lipid chains in lipoproteins. And then through the Dyer experiment, we can cut those right back. We knock at the diffusion, knocks out the glucose. The T2 relaxation knocks out part of the lipids. But this J-edited approach, which actually looks at phase modulation of peaks as well, it attenuates it further because of destructive interference from multiplets, which have got different J coupling constants. So this is a pretty cool experiment because it allows you to go down from all the complexity of plasma to almost the biomarkers that you need that are diagnostic for COVID in one go. And to prove that we could actually translate this is the JEDI spectra that we call, as we call it, on the 600 megahertz machine. And this is from the latest generation 80 megahertz machine. It's a little benchtop machine. And here you can see the GLIC and the SPIC. The GLIC goes up, the SPIC goes down. In fact, it's almost clearer on the 80 than it is on the 60. And if we do this, there's one of those little machines now. So less than $100,000 this machine, as opposed to about 1.8 million for the other. And it has a 0.988 correlation coefficient. So we can take discovery from the big biomarker platforms and translate that back into much smaller and cheaper platforms that you could potentially stick in a hospital ward or even potentially in a doctor's surgery. And finally, I just want to say one thing. So that's what is SPIC, right? So I mentioned it's rather enigmatic, this supramolecular cluster that is present in lipoproteins, but it's phospholipids. In fact, it's quite interesting. We can do various statistics to show what it is, but it links in very strongly with apolipoprotein B100A1, and it's statistically almost equivalent to it. So not only is it a biomarker of COVID, it's a biomarker of the cardiovascular risk. And if we go into a little bit more detail, if we cross-relate the SPIC peaks here, I'm not going to go explain why there are 3 SPIC peaks now, but this is the cross-correlation with the intensity of the phospholipids in the different lipoprotein subcompartments. And these are what we call half-shy diagrams. Left-hand side is control, right-hand side is COVID. And you can see the differential connections there with the different lipoproteins. These are with all the cardiovascular risk markers for the same peaks, including the apolipoprotein B100A1 ratio. And these are the actual mass spec generated lipids, right? So lysophosphatidyl 16-0, for instance. So that's really cool because we now know exactly the molecular composition of the biomarker. And that means we can start to look for the effects of the biomarker in other tissues. And so we've used our Fourier transform mass spectrometry platform to cross-link the biomarkers that we discovered from the lipidomic platform and project those into the moldy FTMS of a carotid artery atherosclerosis model for mice that we happen to have lying around. And there are some of the things that go down in COVID, and they're ending up here and here in the carotid, the coronary arteries of the animals, or in the case of humans, we're working on that now. So I think put together, that shows the power of phenomics, not only as a process for us understanding the origins of disease, but also how diseases which are actively evolving, such as COVID, can be tackled in real time to produce, I think, real clinical solutions and things that will help us understand the mitigation of those in the future. And just the final slide, which I just flashed up accidentally, is that is one of the reasons why I'm in Western Australia. That is outside my house, and it is beautiful here most of the time. But we also have one of the best laboratories in the world now for tackling the sort of problems that we are really interested in doing. So thank you very much for your kind attention, and surfs up, guys. If anybody wants to be a postdoc here, it's a good place to do it. And I thank you also to the people that do this work, and the people who fund us for the research.
Yeah.
So I will now try and disconnect from you. I think I have to release you somehow.
Okay, yeah, thank you very much, Jeremy, for an impressive and very exciting talk in very important research fields. Yeah, I think you convinced us that with all the platforms that you have available, you are up for the challenges that we face today, like, for example, the COVID at the very end with really impressive results in nearly no time. Yeah, do we have any questions from the audience? Yeah. We'll just keep mine and then hand this to you. Yeah. No, it's not. Oh,
it's not on. Let's hope that. There we go. Now you can hear me. Right. God, it does look nice down there. I have a colleague at Curtin and have been doing some happy work in WA over the years. Anyhow, extremely interesting talk. Oh, there we go. Yes, of course, now you can see me rather than the back of my head. Hi. In relation, we know already that people who are vaccinated against COVID and then are nevertheless infected manifest much less severe kind of primary illness, and mortality rates are down and stuff. Do we know anything yet about how that affects long-term outcomes? Do we get less severe long COVID effects, or is it too early to say?
Yeah, it is slightly early to say. I mean, long COVID by definition takes time. But what we can say, I think fairly certainly, is that people who are vaccinated, it doesn't protect you completely, of course. It takes the edge off the severity, and it certainly reduces the lung severity quite a lot. So if you get less lung severity, by definition, you are almost certain to get less long lung effects, there's less lung scarring, and that's quite a significant part of long COVID. But what vaccination does not completely protect you from is all the systemic effects, and that is why we think that people, even if they're vaccinated, can still potentially get new onset diabetes, they can potentially get liver problems. Certainly, they can get the neurological problems and fatigue. And indeed, I mean, the new variant, or relatively new variant, I mean, it was only reported to the WHO on November the 24th, and it's already been right around the world, Omicron. That can certainly reinfect people, even people who have been triple vaccinated and have already had the disease. So it's attenuated, but it can still infect you. And that is why I'm still a little bit worried about your Minister of Health's comments today.
Wouldn't be the first time. Thank you very much.
Okay, thank you. There was a second question? Same question. Okay, good. Yeah. Yeah, then let's stick to COVID for a little bit. I would like to ask you, you've mentioned the changes to the microbiome. And I mean, that's also my main interest in the microbiome metabolism. And you have, what do you think is the main factor that the virus has such an impact on the microbiome?
Well, firstly, as I'm sure most of you are aware, I mean, almost any disease process affects the microbiome. The microbes are almost know before you do. Anything that affects the liver because of the bile secretion, the microbes know about that immediately. And years ago we were doing work on liver toxicity, and the first changes you used to see when you gave a liver toxin were actually microbial metabolite changes, because basically the bugs are pissed off by the fact that the liver is not working properly. So it's not at all surprising to me that microbiome is affected, but there are also some extra interactions going on in the gut, which I don't think we fully understand yet, because the gut, of course, has got the ACE receptors, which the virus binds to. So there's some more complex level interactions going on, but it does look, there's several papers out there now that are showing that there are significant shifts in the microbiome, including things like the Faecalibacterium prausnitzii, difficult one to pronounce, prausnitzii, is a normal microbial species, is actually significantly reduced. And there are other things, there are clostridial species that increase as well. And that is actually a little bit reminiscent of something like autism, where you've got Faecalibacteria down and Clostridia up. So that's sort of fairly worrying to me. What the metabolic consequences of that are in detail, we don't know yet. I mean, what's annoying to me particularly is that about, let's see, when do we start work on this? It was about February 2020. We were trying to get samples in. I was trying to get our clinicians to get fecal samples because I knew this was going to change, but they would not hear about it at all. It was difficult enough to get plasma and urine samples out of them in the early days. So now in long COVID, I think that's our focus now. What is the microbiome change? What does it mean functionally, not just the species change? What are the biochemical changes associated with it, and how does that link into the immunometabolism, which is unquestionably the thing that drives long COVID?
Okay, yeah, thanks. Then I was really impressed about all the platforms that you have available at your institute. And you mentioned that you take from one sample, you run it through all the platforms. So how much sample do you need to really run through your 8 to 10, 12, I tried to count them, it was very hard, platforms?
Okay, that's something else we've been working on as well. I mean, when I was at the Phenome Centre in the UK, we built all our models to run on about 1 mil, right, for NMR and mass spec. We have a wider range of technologies now in the Australian Phenome Centre than in the original NPC. But I was conscious of the fact we used to get pushback all the time from clinicians saying, especially epidemiologists whose samples are very precious, and they want us to work on the minimum amounts of sample. And of course, we're also working on children's COVID now, pediatric COVID, and then you only get very small samples anyway. So we've now managed to modify all our procedures so that we can do the whole lot on 0.2 of a mil.
Okay.
And so about 70 microliters for NMR, and the rest for about 10 to 12 different mass spectrometric assays.
And then, I mean, while the advantages for NMR are absolutely clear, but with all the mass spec analysis, what additional information do you get from the NMR compared to the mass spec analysis?
Well, you get all the lipoproteins because the mass spec destroys them. So we get 112 different quantitative lipoprotein parameters in about 6-minute NMR run. And that's highly validated against centrifugation methods for doing the different lipoproteins. We get a very detailed lipoprotein profile. It comes basically for free with the one-dimensional NMR experiment. But we can also measure multiple small molecules as well, amino acids and organic acids, a whole range of different things. The other reason that we do it is that it actually is sort of, it's a very good quality control check. Remember, in NMR, the sample never touches the analytical system. And in clinical samples, you get quite a lot of bad samples. About 1 to 2% are bad in some way. I mean, they have, they've got hemoglobin in them, because the blood cells have been hemolyzed. They can have solvent contaminants from the swabs and all sorts of stuff. Those things actually don't affect NMR. We can see them, but they don't affect NMR, but they do badly affect the mass spectrometry and basically lower the precision of mass spectrometry. So we use NMR first because apart from the information we get, which is important, we use it to take out the bad samples so the mass spec doesn't get screwed up.
That's fantastic. And thanks for this explanation. Do you have any questions from the online audience? No. Okay. Then I would like to ask you a final question. So your work on the microbiome is really, really impressive. And but if you break it down, just a provocative question: is it all about the diet, or is there more to it?
I wish it were all about diet, because then we could fix bad microbiomes much more easily. What's very important is that there is quite a strong human genomic, or actually a general genomic influence on microbiomes. So I can give you a slightly orthogonal example, which is if you look at laboratory mice and laboratory rats, you can raise them on exactly the same diet, you know, Ratto chips or whatever they call it, and they'll have totally and utterly different microbiomes. And that's because there is a genomic influence on it. So likewise, part of your microbiome is pre-selected by complex interactions with the human genome, because each individual has got slightly different gut environments, which favors the production of certain types of microbiome. But on top of that, that is imprinted by the microbiome that you get from your mother at birth, so the birth canal microbiome, or if it's a C-section, he gets the skin microbiome because of breastfeeding, assuming that the kid is breastfed. The breastfeeding carries nutrients and oligosaccharides which reselect the microbiome. So it's incredibly complicated. And then once you've established, let's call it an adult microbiome, which happens about sort of by the time you're about 3, after that it's reasonably stable, and it takes quite big things to shift that around, like major antibiotic use and things like that. But the diet changes the microbiome mainly not in speciation, but in terms of function. So that although the species proportionalities don't change hugely when you change the diet, what they're doing and how they interact and how they talk to each other metabolically does change. So diet is important. I wish I mean, this is going to be the next 50 years we'll be working, well, I won't be working on it, but people will be working on trying to understand these sorts of interactions.
Especially the interaction of the microbiome metabolism and the human metabolism. I think we can learn quite a lot, as you have shown on one of your studies. Yeah. I'm happy to introduce my colleague Ingela Lannenkopf for her talk now. And I would like to introduce her so she got her PhD at the University of Gothenburg. She then moved to a postdoc to PNNL in Washington State, and then moved to Uppsala, had several positions, including like a promotion to docent in 2017. And very recently, in January 2022, she was promoted to full professor in analytical chemistry. She had several research fundings and also awards like the Berzelius Medal that she was awarded to, and she's also currently the head of analytical chemistry division at our department. She has also been involved in several mentoring projects and programs, and now we look forward to your talk.
Thank you, Daniel, for that kind introduction, and also thank you for the invitation to speak here today. So I want to follow up a little bit on Professor Nicholson's saying about the temperature, and today we have also sunny here in Uppsala, but only 2 degrees, unfortunately, so I'm not sure which is better, 39 or 2, but this is what it is here. So I will also talk about a similar topic, which is metabolomics. So in particular, I will talk about spatial metabolomics, and I call the talk combining the where with the what. And if we look at this figure here, we see that we have a lot of different animals, but where is what animal? Well, you can zoom in and you can see that the bird here, for example, is on the tail of the deer. So this is a way of then showing something very simple, but if we take a little look at something more difficult, that is, for example, the different brain regions. So what are these brain regions, or where are the brain regions? So this has been studied for many, many years with microscopy and other types of visualization tools, and to understand the function and how they correlate these different brain regions and where they are, etc. So we pretty much know that, where we have different brain regions. But what is the chemistry? So the chemistry of these different brain regions will then portray in how they actually function or dysfunction together, because the cells are different in each of these regions, so they have different tasks, and therefore they also have different chemistry. So one typical way that we look into chemistry is to use tissue. So now I'm looking here at a mouse brain tissue. So this is a whole brain of a mouse. And to look at the chemistry, we usually put it in some sort of solvent and make it homogeneous of it. So we take all of the tissue together into one big sample, and we can then do some chemical analysis and figure out what kind of small molecules and large molecules and all of these that we have. And this is great because we get a profile of the chemistry in the brain. But it does not tell us where the chemistry is necessarily. So what can we do then to combine this, what the chemistry is and where it is? Well, I will talk about spatial metabolomics using mass spectrometry imaging here. So in mass spectrometry imaging, we take the tissue, such as a brain tissue, and section it into several thin tissue sections. So these are then about 10 or 12 micrometers thick. We can then probe the tissue on many, many locations all over the section and generate data from each and every one of these pixels. So if we select one pixel, like the red one here, for example, we can look and see what the mass spectrometer has detected at that specific location. So the mass spectrometer detects an intensity of an ion. So on the y-axis, you have the intensity, and on the x-axis, you have the mass over charge. So if we're talking about ions with one charge, we're talking about the mass here, basically. So we can then get this mass spectrum for every single pixel. And the beauty of the whole thing is that if we then select a specific ion, like this one, for example, we can portray it back onto the tissue to figure out where exactly is this ion localized on the tissue. So where did the mass spectrometer detect this specific ion on the tissue? And we can then get a chemical map that can show us where the chemistry is. And we can do this for any ion that we have detected. So we can then see the different localizations of the chemistry in the tissue. So there are several techniques that can do this. One of them, the oldest one, is called SIMS. It stands for Secondary Ion Mass Spectrometry, and it's been around since the 40s. And I think you will hear a lot more about this technique by our next speaker, Professor Ewing. Another technique is called matrix-assisted laser desorption ionization, or MALDI. So while SIMS are using primary ions to hit the specific pixel of the tissue, MALDI is using a laser. And then the ions that are created from either the primary ion or the laser are then transported into the mass spectrometer for detection. Now, MALDI is a bit younger, so it's been around since the 80s, approximately. And this is also the technique that we just saw in Professor Nicholson's talk towards the end, where they had mapped the aorta there. And another technique that we can use is to do liquid extraction of specific locations. So then you're doing a small liquid extraction at a specific pixel, so to speak, and then there are several techniques for this. And the one portrayed here is nanospray desorption electrospray ionization, which was invented in 2010. And since I have worked with this since 2011, I will pick this to continue the discussion today. But I want to point out, so we heard Vice-Rector Professor Tusk yesterday talking about the Nobel Prize. So I want to point out that the Nobel Prize in 2002 was awarded to Koichi Tanaka for MALDI and for John Fendt for electrospray ionization, which is the ionization that is used for nanodesi here. So, in nanodesi mass spectrometry imaging, we put two fused silica capillaries in front of the mass spectrometer. These capillaries are small. They're about 150 micrometers in outer diameter, which is about 2 strains of hair. And we then have a liquid that we push through these capillaries, and the flow rate of this liquid is about 0.5 microliters per minute. So if we translate that, it will take about 100 minutes to generate 1 drop of water, which is very low flow rate. So we push this flow through the first capillary, it then comes down onto the surface where we have the tissue, and it creates a little liquid bridge to the second capillary. So when this liquid bridge touches this tissue, molecules are desorbed into the liquid and then transported further through the other capillary to be electrosprayed into the mass spectrometer and generate ions that can be detected. So to do this, we need to apply a high voltage of 3 kilovolts to the liquid, and we're also using a temperature of about 300 degrees on the inlet of the mass spectrometer. And so then the mass spectrometer can detect specifically what ions that we have on this specific location. But how do we do imaging? Well, we do imaging by moving the sample under this liquid bridge in many consecutive lines, while we're continuously acquiring data. We can then take all this data and put it together in piles, so to speak, for the ions that we're seeing. So here's an example of 4 different phosphatidylcholine species, so these are lipids. And you can look at the images. The darker the color, the less intensity, and the brighter the color, the higher intensity. But what is fascinating with this, if you look at the chemical structure of a phospholipid that is called 38,4, that stands for it has 38 carbons and 4 double bonds. So if we look at the ion images of, for example, 36,1 and 36,4, which is to the left side here, we see that they're very different. But the only thing that actually changes are 3 double bonds. So the 36,4 has 3 more double bonds. That's the only difference in the molecule. And if we're looking at the 36,4 and 38,4, the only thing that differs are 2 carbons. So these small differences are very, very important in biology in the function of these different regions that we have in, for example, the brain. So there, it also shows then with mass spectrometry imaging that we can see where metabolites are localized, and we can try and understand the function of these different regions depending on what chemistry that we have. But with nanodesi, we see lots more than phosphatidylcholines that you have here at the master charge between 700 and 900. We also see a lot of metabolites in the lower mass region. So these are like glucose, creatine, glutamate, amino acids, all the different types of things you can think about almost. And in the middle here, where it seems to be only blank, if we zoom in, we see lysophosphatidylcholines, monoacylglycerols, and diacylglycerols, etc. So what can we use this for? So here is an example of spatial metabolomics from a stroke mouse model. So these are a little bit different colors, but the scheme is the same. The darker blue, the less intensity, and the brighter, the more intensity. And actually here, if you pay close attention, it's not only intensity because these are quantitative scales. So in the stroke model, there is a part of the brain tissue that is damaged by ischemia. So you can see that is circled here with a red circle. And if we take a look at these molecules, we see hardly any difference in the first one, the PC. In the second one, the LPC, we see an increase. And this makes sense because we know that the PLA2 is activated in ischemia, and that generates this molecule. The next one is acetylcholine. We see slight decrease, and GABA is slight increase. So these are neurotransmitters. And if we look at some amino acids, we see a decrease in the glutamate and an increase in the phenylalanine here. So this is a work in progress where we're trying to understand what is happening with the metabolism at the site of ischemia here. But we can also look at, for example, kidney. So this is an example of a rat kidney. So the kidney has 2 major regions, you can say. One that takes the blood in and filtrates it and generates primary urine that is then transported to the center, to the medulla. So if we look at ion images here, we can see the different molecules are present in these different regions, like this, and we can then by that distinguish at least 4 specific different regions of the kidney. So kidney is, there is something called diabetic kidney disease. It's a progressive disease for diabetics, and that the kidney then fails to filtrate very nicely. So it fails to to filtrate away the proteins, etc., from the blood. So if we look at just 2 weeks of a diabetic model, or a model for diabetic kidney disease, 2 weeks after, if we look at microscopy, this is H&E staining, we see hardly any morphological changes. So it looks like they're still healthy. But if we then instead compare the regions from the healthy and diabetic rats with mass spectrometry imaging, we see a great increase in carnitines, this is propionyl carnitine, but also fatty acids like this one, linoleic acid. They're increased in this region where the filtration happens. We can also see an increase in monoacylglycerols and diacylglycerols, and interestingly, specifically in the ones that have acyl chains of 18,1 and 18,2 carbons and double bonds. We also see increases in long and short chain acyl carnitines. So this suggests that the chemistry comes before the morphology changes. So we can see changes in the chemistry before we can see it visually. And specifically, these long-chain acyl carnitines are highly involved in the fatty acid oxidation that also includes the fatty acids. The decrease that we see is in amino acids, so lots of different amino acids. And this is also connected to the long-chain carnitines since they are involved in the TCA cycle precursors and also in the branched amino acid cycles. So overall, what we can tell by this very early stage that we're looking at with mass spectrometry imaging, that we have perturbations in both the fatty acid oxidation in the TCA cycle and an increased amino acid metabolism. We can also see that there are no differences in relocalization of the chemistry, so it actually happens on the site, that the cells themselves are changing the metabolic profiles here. Okay, so there is a limitation on mass spectrometry imaging. And that limitation has to do with that the mass spectrometer detects a mass, which means that isomers that have the same mass cannot be separated. So here, for example, is an ion image of the mass to charge 118. That corresponds to a specific molecular formula, which can correspond to two very different metabolites. Valine, which is an essential amino acid, and betaine, which is a very important osmoprotectant. So which one is it that is actually localized here in the medulla of the kidney? And how can we figure this out? So what we have done in my group to try and probe this is to develop a new technique that we call surface sampling capillary electrophoresis. So the surface sampling is happening in the same way. We have a little liquid bridge that is desorbing molecules from the tissue surface. But instead of then directly infusing it into the mass spectrometer, we have added a capillary for electrophoresis. And by having a high voltage, we have an electrical current that will then drive this flow through the capillary. And then we can separate in the capillary, we can separate the analytes based on their charge and based on their size. So if we take a look at valine and betaine as an example, and we run standards through the capillary electrophoresis, we can see that they migrate at different speed. So valine is a little bit bigger, so it comes out of the column very earlier and is detected by the mass spectrometer, while betaine is a little bit later. So now, if we look at the tissue and we use our new technique of surface sampling capillary electrophoresis, we can then probe the location in the medulla and see what we have there, and we can then probe the location of the cortex and see what is the molecule there. So if we then compare this, we can see that the main contributor to the signal in the medulla is betaine. And which makes sense because this is the osmoprotectant. And the primary urine coming there has very high salt content. So we do need that. So it makes also sense. But we can here detect it analytically. So what else? So what else are there for different types of isomers that we can look into? Well, you probably all heard about saturated fat. So this is a saturated fatty acid. And you probably all also heard about omega-3. So in omega-3 fats, we have a double bond at the position by the third carbon, like here. But we can also have a double bond at position 7 or at position 9. And this is very difficult to tell with mass spectrometry because they have the exact same mass. The only thing that differs is this position of the double bond. And why is that important? Well, because the position of the double bond shows how the orientation in space of the molecule. So these straight fatty acids here, they are saturated. They're just straight lines. While the ones with one bend, they have one double bond. So depending on where the double bond is, the bend will be further up or further down and structurally change the molecule. If we have more double bonds, as you can see in the ones with increased curvature, then you have obviously even more curvature. And this highly affects how the molecules are interacting with each other, with enzymes, with different proteins and everything in the biological system. So how can we then look into these fatty acids with mass spectrometry? So if we can do something that we call fragmentation. And that is you have the mass spectrometer and you hit the molecule of the ion with a high energy, so it breaks. And if we do that a few times here in MS3, we get this profile of small fragments. This is of the oleic acid, which has the double bonded omega-9. If we do the same with vaccinic acid, which is a double bonded omega-7, we see we get exact same fragments. But if you pay close attention, you see that the profile is slightly different, with a slight change in the higher or lower mass fragments that are generated from these. So what we can do is we can then mix these standards in different ratios. So you can see here playing 0, 25, 50, or 75, or 100% of the different analytes here, the fatty acids. And you can see as these are changing, you see slight changes in the profile of these fragments. So since in biology we have very specific ratios of these, we can't say we have just one or just the other. We need to find the ratio. But by eye, it's very difficult to distinguish exactly how much is of each, as you can tell. So what we have done then is that we have done a machine learning tool. We're using a stepwise linear regression model here, and so that we are training it with the data that we get from our standards. On the bottom graph here is the residuals, so you can see that they're very nice and even, and we don't have much of a deviation here. It's about 3 or 4%. So we can then, by using machine learning on mass spectrometry data in MS3 of these fragments, we can see or elucidate the composition of these two specific isomers. But of course, we also want to find out where they are. So if we're looking here at isomeric mapping here, or the spatial, so now we're looking at the ratio of oleic acid to vaccinic acid in the brain tissue, and you can see it's not quite clear, right? So there is some yellow and there is some, so it's not a very clear picture. So let's zoom in a little bit, and we're zooming in on this smaller region here around the hippocampal structure of the brain. So now you have to rethink the colors a little bit. So now the brighter the color, so the more yellow, the more oleic acid. And the darker the color, the more red, the more vaccinic acid. So this is a ratio. So you can see that in the cortex here, for example, we have 79% oleic acid. But in the corpus callosum, we have about 82% oleic acid. So there is more. And if we look at the hippocampus, it's about 74% oleic acid. So there are distinct differences between these brain regions, also down to these very fine isomeric structures. So by that, I want to summarize. So I've talked about spatial metabolomics, and I hope that I've showed you that we can determine the where with the localized surface sampling. We have different techniques for this. And we can also figure out the what with the mass spectrometer. And if we want to go in-depth characterization of what the molecules are, we can do that with either capillary electrophoresis, or we can do it with fragmentation, machine learning. And in addition to this, there are other ways to do it, such as ion mobility, etc., that I have not talked about now. So I want to acknowledge my current and former group members who have all contributed to this work, and also my collaborators for the diabetic kidney disease project here at Uppsala University. Thank you very much for your attention.
Yeah, thank you, Ingela, for a very nice talk with a lot of details in your very nice work. Do we have any questions from the audience? Okay, then I would like to ask you, so you have shown us some quantitative data, so how can you accomplish this?
Yeah, so this is why I very much like this nanodesi setup with the liquid extraction, because that allows us to introduce internal standards into the liquid, and then those are always present at the time ionization. So since if we know the concentration of those, we can then derive the concentration of what we are detecting in the tissue, and we can then get a measure, not only in intensity, but also a measure of how much it actually is there, so we can figure out the differences.
And how stable is the analysis from the nanodesi? Because it looks like a very complicated setup.
Yes, so it takes a little bit of skill. So it's still not a commercial technique like SIMS and MALDI that I discussed here. So we are actually building it ourselves and setting up all of the things ourselves. So you need a bit of training for a few weeks, but then most people get it pretty nicely.
Fantastic. Do you have any questions from the online audience? Okay.
So I'm wondering, Ingela, what is the main limitation? Is it resolution or sensitivity or the handling of so much data? What is?
So you mean spatial metabolomics in general? So in general, I would say that different techniques have different strengths and weaknesses. So some are more sensitive, others are higher spatial resolution, others may do other good things. So I think the limitation is exactly what I talked about here a little bit, to be able to distinguish isomers. That's one limitation. Another one is quantification, because if we don't know what we will detect, it's very hard to quantify it before. So and the data, of course, there is generated a lot of data, but I think we still have some manageable. If you add an ion mobility after that, then it's very, very big, of course. So yes, data can also be a tricky thing, yeah, for sure. But it says with most analytical techniques, if you know what you're looking for, it's always easier.
Okay, and then the final question. So you showed us that you worked on 2 different sample types, like kidney and brain. Which other sample types would be in your interest?
So we're doing all kinds of things. So we've done a few studies using mouse implantation sites. So it's a pregnancy. We're looking into aberrations in lipids and metabolites in pregnancy disorders, so preterm birth, etc. I didn't show that here, but we're also looking into other unique molecules that is very hard to detect with mass spectrometry imaging, such as prostaglandins and now recently steroids as well. So we are expanding the toolbox for all of these.
Okay. Yeah, then I would like to close it down for now and see you later for the panel discussion. Thank you, Ingela.
Thank you.
So now I'm honored to introduce our final speaker of today's morning session. It is Andrew Uring, who who is currently a professor at the University of Gothenburg. He received his PhD at Indiana University, then a postdoctoral work at the University of North Carolina, and then he had several promotions at Penn State University. And in 2010, after some dual professorship at the University of Gothenburg and Penn State University, he moved to Sweden. And just about his accomplishments, he has, for example, 2 ERC advanced grants, which is very impressive. He is fellow of the Chemistry Society, elected to the Swedish Academy of Science, and has like a very strong track record that also led him to take on 2 honorary professorships at 2 prestigious Chinese universities. Now we're looking forward to your talk, and yeah.
Okay, thank you, Daniel, for the invitation, and the committee. Thank you for the invitation to speak at this symposium. I'm going to speak about electrochemical and mass spectrometry imaging of subcellular vesicles. And after listening to Professor Nicholson's talk, I almost changed and wanted to give my talk on COVID in the brain. Okay, but we'll do that another time. Today, I'm going to talk about some research we've been doing to try to understand neurotransmission by looking at vesicles and understanding the vesicle structure and content and how much is released. And I'll show you that in this talk. And so this is just a preview of a few of the slides. I'm going to talk about 2 different techniques, electrochemistry, and and I'm going to talk about mass spectrometry, as Ingela mentioned, just a little bit. I'm going to give you the analytical aspects and then a little bit of the results. Let me see, I'll turn on the laser pointer, see if I can make it advance. So we have developed or used and developed, on the left side is a method that was developed about 30 years ago, to measure exocytosis from single vesicle release at single cells. And it's been used in a variety of different applications, as we'll show. You can put a carbon fiber electrode on top of a cell, stimulate the cell, and measure the contents of vesicles as the vesicles come to the membrane and open up and release their contents, okay? And we can also do this with nanotip electrodes, and we do this for a reason that we use the nanotip electrodes inside of cells as well. The kind of results you get are individual current transients for each exocytosis event, where we're oxidizing at the electrode surface the molecule of interest, which might be dopamine or other molecules we've looked at, which gives a 2-electron transfer. And so to give dopamine orthoquinone, and we can measure this and calculate how much has come out, okay? And you get a current transient that, if you blow it up, has a bit of a complicated shape because it opens a little and then opens some more and then closes, and I'll describe that in a minute. And you get this total charge here, and by Faraday's law, we can calculate the number of moles of molecules that have come out during the exocytosis event. But in 2015, we made 2 different discoveries, and really Johan Duneval made one, who's been for a while at Uppsala, and Xianchen Li and Sudi Maidi did the other, along with Johan working with them. And the culmination of this is that we could put a carbon fiber nanotip electrode inside of a cell, and vesicles will come up and adsorb to the electrode and then open on the surface, and we can measure the contents of single vesicles. When they open, we get the same kind of response, looks like this all over again, and we can again use Faraday's law to quantify these, okay? So the key thing here is that we can compare between the 2 methods to see what is released and what is in the vesicles. And we had done this another way a few years before this, but we wanted to confirm this, and it's still rather controversial in neuroscience. A lot of neuroscientists ignore it. I just reviewed a paper yesterday where they didn't even understand that this took place. And so 70 years ago, Bernard Katz proposed that basically when a vesicle comes up to the surface, everything is released, and And that's what they call the all or none principle of exocytosis. And the interpretation of it is incorrect. Okay, it's not correct. When we look at this more carefully, and this was first done at PC12 cells, and I'll show you some more. Exocytosis, we were measuring 73,000 molecules coming out in this set of cells, and cytometry in the same set of cells was for 114,000 molecules. So about 63% was coming out during this release event. And so we realized this had to be partial. And really at the same time that we did this back in the day, Christian Amator's group in France modeled this dropping here and said this can't be due to diffusion and using up the material. It has to be due to closing of the vesicle again. So we sort of co-discovered this. Now we've come up with methods to look at this simultaneously on the same cell. We use nanotip electrode on the outside and nanotip on the inside, and we can actually oxidize then material as it's being exocytosed and oxidize material in vesicles and compare them. And here now we did the IVIEC, that is what we call the intracellular vesicle impact electrochemical cytometry. That's the inside. Before the stimulation, before we stimulated the cell to undergo exocytosis, and then during exocytosis, we measured the release, which we call SCA, or single cell amperometry. So this is what we're looking on the outside. And then we looked at the IVIEC after the stimulation was over. And now you see a few things. First, only about 63 or 60% comes out in this case, okay, than what we see in the vesicles. And after exocytosis, we see less in the vesicles. And there's actually a complex change in the time. I took it out because it's too long to explain. But it does go down. And that's because we've used up some of the material in some of the vesicles. It doesn't go down as much because we don't release from all the vesicles in the cell, only from some. So this is exactly what we expect, and we could even calculate a percentage, a rough percentage of how many vesicles were undergoing release, which is about 30%. Okay, and so in that area that we're measuring, and that calculation can be is a large range. Okay, but we can look at these things at the same time. So we took methods like this, basically this type of method, and we looked at a series of cells. The cells I just talked about were PC12 cells. We've also looked at adrenochromaffin cells, and we see in the range of about 58 to 74% or 60% released, we'll say. We've looked at serotonin that's loaded into pancreatic beta cells where normally insulin is released along with serotonin. We can't measure the insulin. The electrodes that have been reported to measure insulin don't work in our hands or actually the hands of anybody else at the moment, but we could see the relative amount of serotonin coming out, and it's about 34%. And we looked at Drosophila neuromuscular junction neurons, and we got really low. Neurons are really low, it looks like. We looked at human neuroprogenitor cells, and this we haven't published yet. Preliminary data, it's 8 to 22%, sort of in that same ballpark. We looked at neurons from the gut, okay, so back to the biome, okay, this is kind of cool. We looked at bone cells, and we see 80%, and we can treat them with certain drugs and get them almost to 100% release. It's kind of fascinating. And this Christian Amator has been on and off collaborator of ours with Weihua Huang in China, looked and found 30 to 50% released from glutamate neurons with a biosensor they use. So this is a phenomenon that is pretty universal, it looks like, at least cells that we're looking at, and at least for amperometric detection. And so I'm going to go past that. Here's an EM of a cell, nucleus in the middle, a lot of vesicles here. Okay, just to give you the idea, we put the electrode on and we look, and we've come up with 3 different modes of exocytosis. This mode and this mode were there before us. Now partial release or open and closed, sometimes we call it. Partial release is in the middle. Now, this particular release we find is at least 95% of the time when we're looking at our cell systems that we're looking at. And so you see some full release, and there's a method we use to try to estimate that, but it's probably 2, maximum 5% of the time. Most of it's this, and this kiss and run, we haven't really formulated how often this happens, but it does happen. Okay, so the key thing here is that partial release makes the closing of the vesicle an important part of the regulation of individual events and synaptic strength. In terms of individual events, in terms of overall events, of course, the number of events and the snare complex and how it affects it is important. But in terms of the plasticity from individual events, it's the closing of the vesicle that's going to make a difference because it allows more or less transmitter come out. So we've had a lot of flack for this because we only had one method to show this. So we went and we used nanosims. And we've been working with this for a few years now. We have the nanosims in Scandinavia. And what we did in this technique, if we go to the right first, is we take our sample and we shoot ions at the surface. This is the SIMS that Ingela was alluding to. And the ions hit the surface and sputter off ions and neutrals, but we won't worry about those. In nanosims, we really blast the surface, so we basically clean it up. It's a pretty destructive technique. It breaks the ions down to their primary atoms, and some of them recombine to give you dual atom, or small biatomic molecules, okay, if you want to call them that. And that's what we analyze. And these are come up here, and we pass these because they're charged through a magnetic sector turn and then a magnetic sector analysis chamber. And we can analyze 7 different molecular weights at once. Okay, so this is how we do this. And the cool thing about nanosims is that we can do it coaxially, and so we can get really close to the sample and get small, a very good spatial resolution. So our spatial lateral resolution is down to about 50 to 100 nanometers. Depth resolution, if we profile through, is about 20 nanometers. So we can look at vesicles this way in really high resolution. And the precision in the vesicle volume is about 10%, okay, the carbon-13 precision. So, okay, so what we did in this technique is we took L-DOPA, which is the precursor biologically to dopamine. We bought it labeled with C13, so we isotopically labeled it. And then we put this, we incubate our cells with L-DOPA, and the L-DOPA goes in and via dopa decarboxylase loses one label but gives you the dopamine with 6 labels on it. This dopamine is then pumped via the vesicle monoamine transporter into the vesicles and starts to fill up with dopamine that is labeled. Now remember, there's already dopamine there that's unlabeled. So we can look at the C13 to the C12 ratio here and figure out how much this is. And this is nice because we have a background in the C12 and we have the C13 here. And a whole nother talk I could give, we've actually come up with a way to quantify molarity of the dopamine in the cell, in the vesicles, okay, and we published that recently. So we take the cells, we dehydrate and embed them, we do TEM imaging to see where everything is, and then we put it in the nanosims for imaging. And when we do this, we get images like this. This is a few years old now, lead in. But here's the EM of the cell, so we can see where the vesicles are, these places with the dark, dense dots in them. That's the protein core in the vesicles. These vesicles are, this is a cell that's been loaded with L-DOPA, so we could put in the C13 L-DOPA, and it's loaded to where, so the vesicles expand, something we discovered 22 years ago. And so the vesicles expand when you load them up a little bit, but they're about 200 to 300 nanometers in size. So there's little dots here, okay? And then when we do the image, this is the C12 image, this is the C13 over the C12. There's a lot of carbon there, of course, so there's a big background. We can subtract it out and we see the vesicles, okay, here. Now we can blow these up. Our resolution's way better. We just wanted to see the whole cell. So here's a blow up. Here's the blow up of the TEM showing the dense core, the proteins and the halo around it. And here's the signal for the C13 over the C12 in the nanosims, okay? And I should point out this is from here and this is from here. And then if we incubate for a long time, okay, longer time, instead of 1.5 hours, 12 hours, we find that the signal, which is started out mostly in the halo here, mostly here, moves to everywhere, dense core and the halo. And this is a different set of cells, a vesicle from a different set of cells, of course, because it's a longer incubation. But we see that it spreads out, and we did this in a number of cases. So it's kind of interesting. We can start to look at the subvesicular content. Well, we wanted to take this technique and look at partial release. Could we show partial release? And Lisa Melander, along with, came up with a brilliant idea. She was a postdoc in my group for a while. And Ter and Mai worked on this and really worked out all the details. And what they did is they took C13 L-DOPA and labeled and incubated cells, and this goes into the vesicles as dopamine, as I described. Then they stimulated the cells with potassium to undergo exocytosis in the presence of this DEA, this drug that's labeled up here. It's a drug with an iodine, a natural I-127 in it. Iodine is a really good signal, gives a really good signal on the nanosims, so we can detect it very nicely with the nanosims. And so that's why we use this. Now, when we stimulate the cell, it undergoes exocytosis, and if it's partial, some of the dopamine should be coming out. So this is what we should see losing, and that's going out. And then some of the IDEA should go in, okay, while the vesicle pore is open. And so we should see iodine going into the vesicles. So we looked for this. Now here is the experimental data. Here are TEMs, okay, and with just stimulation and then stimulation with the iodine, okay, with the DEA, okay, with the DEA down here. And then this is looking at the C13 over C12, so the L-DOPA, and this is looking at the I-127, so the DEA, the iodine labeled DEA. It's naturally labeled. It's normally there. I call it labeled. Don't want to confuse anyone. And then when we looked at the data down here, and we can look at the number of molecules, okay, coming across that we're measuring in these things, and plot them as a function of the intensity of this, okay. And so now here, this is the ratio of the 127 over the C13 here, and I'm going to blow this up in a minute. And this is the rate, the C13 here, okay, amount. And you can see, and then here's the plot from all of these different vesicles that are plotted here along here. And let me just put this up here. This is make this axis a little bit bigger so everyone can see it. Here's the control, which is down at the bottom. You don't see anything because there's no C13 and no iodine. Okay. Then we have C13 with no stimulation, and that's the red down here. Okay. And then we have that's the largest. And then we have that's here red. And then we have C13 L-DOPA with the stimulation, and we see it changes. And then we have the C13 L-DOPA in the presence of the iodine when the DEA. Now, the best chart to look at is this part here, which I've left up, but I'm going to expand it from here and here to here. And what you see is that when we have no C13 label, we see nothing. Okay. If we have no C13 label but no stimulation, then this is the amount of C13 we see, the signal that we see in the vesicles, okay, on average. If we stimulate with potassium, well, this is what we see. It drops, and it drops about I'll show you the quantification in a minute because we have a way to quantify this. And then if we do it in the presence of DEA, it drops a little bit more, okay? There is a difference here. And so we think that DEA, and we've done electrochemistry to show that DEA changes slightly the amount that's released, okay? And that's important. But we see the drop here, and that's the main thing. And the big thing we're looking for, of course, is does the DEA go into the cells? And here's no C13 L-DOPA. Here's no stimulation with the C13 L-DOPA, looking at the DEA. Here's if we stimulate, a little bit of something's happening. So there's a little bit of background iodine we see. But then there's a huge increase in the iodine that we see with the DEA present during the stimulation. It's very large, okay? So can we quantify this? Can we calculate the fraction released? Okay, and here's the number of molecules that we see. Here's none. Here's after potassium. Here's after potassium with DEA. Okay, looking at the number of molecules, looking at just the L-DOPA, and so this is a pretty complicated equation, but we have a background. I'll just say quickly that we have a background that consists of the resin, and the resin gives us a carbon molarity of about 50 molar, and so we can actually use this as our background to come up with a concentration, because we know what the concentration of the background carbon is. And we can come up with the numbers here, but in this case we're doing the fraction, so we get the concentration, and we have the vesicle stimulated and non-stimulated here, okay? And we find that it's about 60%. It's right around 60%. So that's fascinating to us, okay? That's exactly what we predicted from the electrochemistry experiments. Now, we're interested in the brain and memory, okay? And if you look at partial release, it's important because if you have 2 times the release events, which is the original idea, and this is true too, this still works, okay? But if you have 2 times the release events, you get 2 times the synaptic concentration. And this is where the snare regulates the frequency of release events. But if you have a 5% release and you change it to a 10% fraction released, okay, for the same number of rents, you get the same effect. Okay, so a 0.05 fractional change in release leads to a doubling of the transmission efficiency. Even starting at 20% would mean a 25% transmission effect. It's huge, okay, this idea of fraction released. Now, we can dissect vesicles. We can start to dissect vesicles, and I showed you this already with the nanosims. But I'll also point out something we just came out, I think yesterday or the day before in Agavante Early View, okay, where we can actually look at dynamically with electrochemistry. So I showed you the nanosims, but what Shulan He and my group has discovered, she's been looking at Hofmeister series ions, and the chaotropes is cosmotropes and chaotropes, and the chaotropes disrupt a lot of the biological structure, okay, and cosmotropes make it more rigid. So what she's done is she's come up with a way to disrupt some of the structure, and when she puts a counter ion to the stimulant, to the potassium stimulant, she puts in potassium and then thiocyanate is a chaotrope. Okay, what she finds is that she gets 2 peaks, and this is very consistent. I have a lot of these now. And these 2 peaks, she's worked out, this one is due to material being released from the halo around the dense core, and this one is the material being released from the dense core, which takes longer, which has been made, which has been disrupted by the chaotrope. And what she finds is that there's a little bit more material, as we expected from other measurements indirectly in the dense core, but not a lot, a little bit. The fraction, though, here's the fraction of each. Here's the fraction in 1. It's about 30%, and the fraction in 2 is 70%. So I say not a lot, but it's some. Okay, and so here is what happens if you use chloride, which is a cosmotrope. You get a single peak. Here's what happens if you use thiocyanate. Okay, and you get 2 peaks. This is a hand drawn. Okay, that's my drawing. But then you can look at the halo and the dense core independently, and this is really interesting because we can start to tease apart concentrations in 2 different methods to confirm each other in these analyses. Okay, so IVIC and SCA allow direct comparison of exocytosis and vesicle content. Exocytosis seems to be predominantly partial in nature. Small fraction of transmitter is released during exocytosis in singling neurons. Exocytosis is predominantly partial, confirmed by nanosims, and these are the people who did all the work that I've been talking about. These are some of my collaborators down here on some of these things. These are people I mentioned in the talk, and this is a few months old picture now. A couple of people have left, more people coming on the bioanalytical imaging and sensors groups at University of Gothenburg. And I thank you for your attention.
Yeah, thank you very much for a very nice and impressive talk covering some of your work in analytical chemistry. Do we have any questions from the audience? Okay, then I have a question about your dopamine oxidation that you have shown us at the start. How selective is it?
Can you just say that again? How selective is what?
How selective is the oxidation of dopamine so that you detect?
When we're doing amperometry, anything that oxidizes at that potential will oxidize. But we also can scan, and then we get reasonably good selectivity. Dopamine, norepinephrine, epinephrine, unless you do some tricks with the voltammetry, all look about the same. But we can, we've chosen cells that only release dopamine, or in the case of PC12 cells, maybe 10% norepinephrine, but not in our hands when we've done analysis of them. But some people say that. But the key thing isn't really the selectivity in that particular case. It's the amount coming out, and whether it's dopamine or 10% of its norepinephrine doesn't matter because we'll oxidize it the same. So we have selectivity when we need it, but we don't need it in this case, so we don't use it. That's for the electrochemistry. I think that's what you're asking about. For the mass spectrometry, of course, we're very selective. We know exactly what we're measuring.
Okay, perfect, thanks. And then you have described the vesicles. How large are they?
Yeah, the vesicles in the PC12 cells, for example, are when we incubate with L-DOPA, probably around 200 to 300 nanometers in diameter. They're normally about 180 nanometers. The vesicles in the cells, and let's say the neurons, are somewhere around 50 to 80 nanometers in diameter.
Okay, yeah, thanks so much. Yeah, and your quantification, I mean, with the carbon-13 labeled dopamine that you use, can you also apply it to investigate other molecules in your system?
Yes, we're measuring. I think, if I understand the question, you're asking if we can use the quantification to measure other molecules besides dopamine. Is that correct?
Yes, that's
Yeah.
correct. Yeah.
Okay, so I have. Yes. I mean, actually, I showed molecules. In the adrenal cells, we measure norepinephrine and epinephrine. In the other cell, the PC12 cells, we're measuring dopamine. In the fly neurons, we're measuring octopamine. It's a bit different molecule, but similar sounding name. We measure serotonin in the beta cells, where we can measure anything that's electroactive, and if you look at the paper that Amator and Wang published, they look at glutamate with a biosensor. It's a really special biosensor because it goes super fast. And somehow is quantitative counting wise. I haven't quite figured out how yet, but I'm convinced it's right. And so we can look at a number of molecules, and more are being developed, of course, for things that need biosensors. But I would say you can look at dopamine, norepinephrine, epinephrine, serotonin, histamine, which I didn't put up here, serotonin, and glutamate so far. So.
Yeah, really nice. Do you have any questions from the online audience? Okay, yeah.
I can't see the audience, so I can't see you. I'm just seeing my screens.
I am. So my question is, do you have an idea why exocytosis is partial? What function does it have?
Yeah, that's just what I tried to say in that slide, Mata, and I can hear who is talking. I can't see any of you guys. I'm not sure whether I'm in the wrong view or. Let me see. No, I'm not. Okay. You guys took that away from me somehow. But let me. The point of partial exocytosis, okay, is that it regulates plasticity, okay, and it's if you look, we did an experiment where we looked at fraction released with cocaine and methylphenidate. And cocaine is a drug that is you're stupid if you take it, but you're more stupid after you take it, okay? It diminishes cognition, and methylphenidate improves cognition. And what we found is that the fraction released as you went up in cocaine, okay, went down, and the fraction release goes up with methylphenidate. And we've since looked at a number of drugs, like student study drugs and things that are to help to improve cognition, and drugs that we know diminish cognition, like anesthetics and so forth. And it always is the same trend. The fraction released is related to things that have to do with cognition. So we're trying to develop tools to look for the mechanism that goes between fraction release, how much is released, and then the downstream memory formation, because we're obviously hindering memory formation as we decrease the fraction released.
Thanks a lot.
Okay, so if there are no more questions, there's one more. Yeah.
Hello, thank you very much for the talk. My name is Robin. I'm a PhD student here at Uppsala University, and I'm curious about the oxidation of this dopamine. You said basically you not only oxidizing dopamine, but everything which would be in this area of possibly oxidation.
Yeah.
What are you reducing at the same time?
Oh, well, I mean, reduction is taking place at the counter, at the reference electrode, okay, the counter/reference electrode. It's a very small level. The currents are so small that we don't use both a counter and a reference electrode. And the whatever you're reducing is whatever easiest to reduce at that electrode, but it's at a different place. It's not at the cell. So you're oxidizing the dopamine and you're generating electrons that have, generating protons that have to diffuse away. So.
But then the two electrodes have to be quite close to each other, else you have a huge resistance, I can imagine. So over time you change your system.
No, they don't have to be close to each other at all. I don't follow you on that. Why would they have to be close together? It's just a current flow. It's an ionic current. It flows very quickly.
Okay.
Yeah, no, it's just an ionic a proton current. So as you make protons, they have to move away. Protons have a very high diffusion coefficient. And actually, if you look carefully, we've been studying this. You're leading me into something maybe inadvertently that's interesting. We've been studying the effect of pH on this process of opening of vesicles on electrodes. And it turns out that protons move along a membrane at different rates based on the pH. And we're seeing this. We're seeing differences in this because of these, probably because we're making lots of protons. If you oxidize, just to whet your interest a little bit, if you oxidize dopamine in a adrenal cell vesicle, there's 2 million molecules, and 2 protons per molecule means you have 4 million molecules happening at a pore that's open only a few nanometers, okay, flooding out, and things flooding out in 80 microseconds. So you're generating, what would normally be something like a proton bomb, okay, but they shoot along the membrane really fast. So it would be a nanoscopic proton bomb, but they shoot along the membrane very fast. So they escape, and we can actually, we're starting to find out how to actually, I wouldn't call it regulate it, but to change that.
Okay, thank you.
You're welcome. Good question.
Okay, yeah, thank you very much. Then we will close down this talk. Thank you, Andrew. And I would like to ask Ingela to come to the stage that we can start the panel discussion. So yeah, so we've heard 3 talks that can be summarized as analytical chemistry on the field of analytical chemistry. So this was the combination, really impressive work in different fields. So I just want to ask you in general, because since the chemistry combines you, what is the importance of the chemistry research in today's society and also scientific community, especially that we move more and more into interdisciplinary research?
Do you want to ask me specifically or just general to the panel?
It's general to the panel, but anyone, yeah, maybe Ingela, you can start and then we can ask, yeah.
I think chemistry is involved in everything, everywhere, in all the products that we do, in food, like we've heard, in our brains, like we heard Andy discussing now very specifically. And so I think it's very relevant also in everyday life.
And Andrew, what is your opinion?
Well, come on. Chemistry is the central science. And the issues, if you look at the issues of the 21st century, you have environment, healthcare, energy, and food supply and materials, right? And maybe utilization of information and utilization of technology. And chemistry is involved in basically all of those.
And Jeremy?
I think Andrew just said it all. Yeah, chemistry is the emperor of the physical sciences. It's where they all meet. Of course, from my own particular point of view, the interface with medicine is likely to be where most of the many key advances will go on in the future. But also others, things to do with energy supply, clean energy, electrochemistry of batteries. things have changed very rapidly with lithium batteries. There's going to be other advances, in fuel cell design over the next few years, which will revolutionize lots of aspects of transport. So chemistry is key to pretty much everything.
And if we then take a close look at analytical chemistry, maybe the development over the past 10 to 20 years that we have all witnessed, and also the future of analytical chemistry. Whoever wants to take it on, please comment.
Well, I'll say a couple of things. Things have changed remarkably in my lifetime. Things that you would have thought was utterly impossible 20, 25 years ago that become completely routine now. And I think there's a great deal of the work that we do is obviously very high-end stuff. It uses very expensive complex technology, complex theory. I think that the big challenges for chemistry and analytical chemistry in the future is not only to make the discoveries, but also to translate those into the real world, to take whatever is in the big laboratory and make it useful whether it's in the patient's bedside or whatever environment it happens to be, or even in analytics for food supplies and things like that. There is an enormous number of technologies available now. Many of them spectroscopic, but a lot of them electrochemical. And I think it's the making those smaller, more sensitive, faster and cheaper, I think is where a lot of work will go on in the near future.
Yeah, thanks so much. Really good thoughts. Anyone else?
Yeah, so I just want to agree. I think analytical chemistry is really a core topic or subject of many different disciplines. So we have it as in medicine, we have it in biology, you have it in the production of food or whatever we eat, or cars we make so that they will be sustainable for a long time. You have it in almost everything analytical chemistry is used. The research in analytical chemistry to coming up with new ways, seeing new molecules, smaller things like Andy was discussing, like pushing the limits. I think that is the research of analytical chemistry, but the application is enormously wide.
Yeah,
let me just add though, because to both if you combine the two, right? I want to point out, it's analytical chemistry is used in all these different areas, and Ingela is completely right. And there are a lot of new directions and miniaturization and new things that need to be done, as Jeremy said. What the rest of chemistry needs to realize is that without this, they don't have these tools that they're using all the time. Because I constantly have, for over the years, I've had organic and physical and inorganic chemists tell me, oh, well, analytical chemistry, yeah, we use analytical chemistry, we're analytical chemists too, or what good is it, to do new analytical chemistry? Look at the tools if you're an organic or inorganic or physical chemist, especially the inorganic and organic. Look at the tools you had 20 years ago and the tools you have now, and then realize how much of that came from there. That's what you're throwing away if you don't push analytical chemistry. And we, that's the negative side. But the positive side is that we're really there trying to push the limits, as Ingela said, and help move these things and give you tools that you can use as well as discover our own science.
It's all great.
I'm glad you, sorry, I just want to answer that. I'm glad you said that, because it was sort of almost next on my list. And in the UK, certainly, if you're not, it used to be, if you're not organic, inorganic, or physical, then you're nobody in chemistry. You've got a hump on your back, right? And yet, so much of all the other fields of chemistry is totally enabled by analytical chemistry. But it's still, and it depends. Different countries have got different attitudes. In the UK, it's got better, but it's still not there with the others. And of course, I call myself a biological chemist now, because that's much more respectable than an analytical chemist. But in Germany, for instance, analytical chemistry has always been much more highly respected than it is in the UK. Same, I think, in America.
Yeah.
I just want to add that I think it's also a little bit sad to see that analytical chemistry is pushed away from being their own subject in many universities into being included maybe instead in biology or something else, just because we're looking at biological samples, and maybe that's where the focus is, to push some of the limits. and so that I hope that trend will stop.
It's a long-term mistake.
Yeah, all great points. Thanks so much. Yeah. So since we have the 300-year anniversary of Celsius weather statistics, how can you relate, or can you somehow relate your research to Celsius work like about 300 years ago? Maybe let's start with Andrew.
I'm not an astronomer, okay? I mean, he did come up with the temperature scale, and we all measure temperatures, okay? But I mean, really the general way to relate, of course, is that he was a pioneering scientist, and we would all like to be pioneering scientists. But I mean, his specific work is hard to relate to. So,
well, I think there is a general application, of course, which is we all require knowledge of temperature to understand chemical reactions and thermodynamics. So there is to be able to measure it accurately or precisely is really important. I mean, I have a particular interest in temperature, although Kelvin might be more appropriate for me with superconducting magnets, because of course, you know, these machines run at very, very low Kelvin temperatures. But just out of interest, the little NMR machine I showed, the new 80 megahertz machine, which, I mean, it's quite interesting. I've spent the whole of my life going up and up and up in field. I find it strange that I'm now quite excited by a low field NMR machine, but that is actually enabled by these incredible new alloys that have been, I think a lot of them developed for loudspeakers. So you now have a wacky great big, enormously powerful loudspeaker that's really, really small. It's the same sort of alloys that allow you to have a permanent magnet at the core of this. But the thing about magnetism, permanent magnets, is that the magnetic field is extremely temperature dependent. So whereas you can plug these little NMR spectrometers in and they will work, they don't need cryogens or anything, you do actually have to let them equilibrate for 2 days. It has very accurate temperature controls, and it has to be accurate to 0.01 Celsius all the time, otherwise the field moves all over the place. So that's an interesting spin from my point of view.
Yeah, and just to add to that, I think also Celsius pioneered tracking the weather, right, and writing down that. And I think for us as mass spectrometrists, humidity is an important aspect of things. So in the dry seasons here, we don't maybe have as much problems as if we were in a country, maybe in Australia, I don't know. Maybe you need to build some specialty rooms to decrease the humidity to have a, good analysis. So.
Yeah, I think that all makes sense, and very good that we can all relate somehow to Celsius work like several hundred years ago. Absolutely, yeah, please feel free. Yeah.
Thanks very much. Yeah, it was very much on board with what you're all saying, but I think there is actually a broader frame than that too, because if you think about where Celsius fits into the frame here of the history of science, he's at the cusp of the transition from sort of generic observation to precision measurement. Precision measurement to temperature being just about the first thing that's established, good equipment, standardized scales. And that completely is the basis for everything that all of you guys are doing. Temperature was perhaps the most accessible thing there, and I mean that around the same time people are starting to look at air pressure and so on in similar ways. But it all leads on from there right up to the kind of completely precision measurement dominated science that we all do today.
Yeah, very good point, I think.
Yeah, and then so we are moving into the age of artificial intelligence. How do you foresee that artificial intelligence can improve your research or take your research into the next level?
Well, I don't mind starting off on that. I mean, artificial intelligence, well, of course, it's been around for a long time. There's nothing really new about it, other than the fact that computers have got a lot faster. But things that I was doing 30 years ago in computer pattern recognition and multivariate statistics would, if I were publishing now, you would probably call it artificial intelligence. So there's nothing new about it other than the fact that it is quicker and more efficient. But any sort of computational activity where you're trying to take a big data and then extract from some useful information or rules, it's only as good as the data itself, and actually in many cases it's only as good as the questions that are asked of those data. If the data is imperfect in some ways, and that's true of many big data sets, or the question is not the right sort of question that isn't reflected in the data, then you haven't got artificial intelligence, you've got artificial stupidity. And there's really quite a lot of papers out there that are not advancing the field at all. So it's a mixed bag. There are some things you can do that are absolutely awesome with artificial intelligence, and there are other stuff which is pretty dumb.
Yeah, I would agree with that. I think actually the place that artificial intelligence is going to become important, and I keep waiting for it, and maybe it's still a little bit too far in the future, but I think eventually with the massive amount of literature we're building up, and the difficulty in looking at that literature to discern things. I mean, in this COVID time, with all these meta-analyses that are being so poorly done, with individuals digging through and picking out from 1,800 papers, 25 papers or 30 papers that they think are the right ones, but for absolutely obscene reasons. Okay, and anybody who knows what's going on knows what I'm talking about on that one. But I mean, what eventually this is going to do is it's going to allow us to plow through the literature and in a way that will allow us to integrate huge amounts of data that our mind couldn't do in any reasonable time. And I think the thing that will eventually do, and it scares publishers and maybe is holding off some development, is that it will change completely the way we put out our data in our publications. Because instead of putting them out with this abstract introduction, etc., you can completely change the format to a more data-driven publication. Okay, and I think this eventually will be the rule. Although 10 years ago I thought this would be here by now, and it's not. So I was off on my timing. But I think within 5 or 10 years we'll at least see some possible part of that. Because I'm sure that in China they're working on trying to develop this. Okay, they want to be a step ahead, and I know people there that are working on it. But I just don't know how far they've gotten.
Yeah, I mean, there's a lot of worries in this for me. I mean, if you take it to an extreme, then you could actually have computers and programs that are capable of sorting data and writing the paper for you, and you just stick your name on it at the end. I mean, actually, that may well have already happened now. So I think it is very much a mixed bag. There isn't also something you just said about publishing. And I noticed there's a question about that a little bit later on. But the thing is, I remember a few years ago there was a controversy. I can't remember exactly. It was a Korean scientist who faked some of his genetic results, and there was a big outcry. It was in Nature, and I wrote a letter to Nature, which actually got published, basically about the fact that scientists are not prepared, they're not intellectually prepared to deal with something that's deliberately fraudulent. We're honest people mainly, and we expect other people to be like that, and we're not really prepared for that. But if you look at the average paper in Nature or Science, it may only be a few printed pages long, but you have a look at the supplementary data, right, and it can be 70, 80 pages of dense stuff, which took 20 postdocs 4 years to actually generate, right? And it's very, very difficult to plow through that as a referee, even if you had the time to do it, and actually come up with an answer as to whether or not that is correct. So in the future, I think artificial intelligence solutions could be very important for helping sort out these very complex data sets that go with complex papers, in order to help the editors and referees sort out some stuff that is maybe fraudulent or just plain wrong.
Yeah, what I'm thinking is that there may be one thing that would be hard though, and that is to sort out. So, I mean, if we want good data, we need good samples, right? So, and Jeremy, I think you touched on that also. And how is that possible? So if we only have, say, robots generating the data and then AI looking at the data, how will we even, know what's good and correct and not?
You won't.
Yeah, these are fantastic points. And yeah, I think we should be careful about artificial intelligence, but use it in the right way. And that brings me also to the next point, exactly about the open data. So where do you think we are standing at this point already, and which steps do we need to take to have open data that can then be in the future be harvested or investigated in more detail by these artificial intelligence softwares that are being developed?
Well, I approve of open access publishing. I think it's a jolly good idea, but I don't like having to pay $10,000, $5,000 to $10,000 for a paper. So it'd be great if it's all free. I mean, I think it costs $10,000 to publish in Nature now. I mean, there's a sort of special selection pressure that exerts as well. I mean, there are people and groups that might be doing good work, but actually can't get it published because of that. But going back to the question, open access publishing, everybody should be able to see everything as far as possible.
But how should we deal with the costs? Because I mean, the funding agencies don't provide more funding to us. So how should it be? Yeah, especially, I mean, you mentioned like the 10,000 US dollars for a Nature publication. I mean, this is like, yeah, of course, it's hard to.
Yeah, but I noticed they don't pay the referees anything. Right, they charge large amounts of money, but they don't pay referees. It's something not quite right about that.
On the other hand, for Nature paper, you also put in a lot of money to. I mean, as you mentioned, like 20 postdocs for years. So then the 10,000 is at the lower end if you can get the data together. However, I mean, of course, the costs are definitely way too high. But I mean, what would be an option for the scientific community to counter this?
Yeah. Well, it's a delicate balance, right? I mean, because as the price goes up, eventually there'll be pressures to push away from this. And many institutions, I don't know about most, but many have started to pay the price for open access to at least to certain deals with certain publishers. And I just keep waiting for the administrations of these institutions to realize how much of their budget they're putting into this publication, when if they all teamed up, they could do more archiving like the mathematicians and the physicists have started to do. And eventually we'll go that way. Right now there's so much pressure to be in prestigious journals that it hasn't taken place yet, but eventually it will go that way. I think open data, there's also sharing of data. It becomes really, it depends on the area you're in, but to put all of your data from a paper, on every paper, onto a server is very difficult, and that's where we're moving. I've gotten away so far with making sure I keep all the data and putting a statement in that it's completely available, upon a reasonable request. But the direction from the publishing, and I know at the American Chemical Society, the direction is to get everybody to put all their data on the server. And of course, that's pushing towards AI, because then an artificial intelligence system could go in and just search for all this data and would not have any need for the publication. The data would be there. So I mean, the future is going to be different. I guarantee you. I don't know exactly what's going to happen, but I can see that this combination is going to play a big role.
The interesting thing also is then you have a lot of server space, and if we listened to climate change yesterday, and then we have all these servers using a lot of electricity and other resources just to keep the data up somewhere for someone potentially to look at it. I mean, I find that it's hard to look at data from some of my group members because I don't know exactly what they did to generate the data, and then I can only imagine what the future might be like.
Yeah.
Okay. I just take a look at the time. We are unfortunately at the end. Very nice discussion. Thank you very much, everyone, for being here. And so, yeah, I unfortunately